Welcome. In this lecture, we will discuss erythropoiesis, the process responsible for the production of red blood cells. As medical laboratory science students, understanding erythropoiesis is essential for interpreting hematology results and recognizing disorders of red blood cell production. What is erythropoiesis? Erythropoiesis is the biological process by which red blood cells are produced. It occurs primarily in the bone marrow and is a highly active process, producing approximately 200 billion red blood cells each day. The main purpose of erythropoiesis is to maintain an adequate red cell mass to ensure proper oxygen delivery to tissues. Sites of erythropoiesis. The site of erythropoiesis changes throughout life. During embryonic development, red blood cell production begins in the yolk sac. In the fetal period, the liver and spleen become the primary sites. During childhood, erythropoiesis occurs mainly in the long bones. In adults, it is restricted to the axial skeleton, including the sternum, ribs, pelvis, and vertebrae. Origin of erythroid cells. All red blood cells originate from hematopoietic stem cells located in the bone marrow. These stem cells differentiate into common myeloid progenitor cells, which then commit to the erythroid lineage. This process is regulated by several growth factors, with erythropoietin playing the most important role. Stages of erythroid development. Erythropoiesis occurs through a series of well-defined stages. These include the pronormoblast, basophilic normoblast, polychromatic normoblast, orthochromatic normoblast, reticulocyte, and finally the mature erythrocyte. Each stage is characterized by specific morphological changes that are important for laboratory identification. Morphological changes during maturation. As erythroid cells mature, they progressively decrease in size. The nucleus becomes smaller, chromatin becomes more condensed, and eventually the nucleus is expelled from the cell. At the same time, hemoglobin synthesis increases, causing the cytoplasm to change in color from blue to pink. Reticulocytes. Reticulocytes are immature red blood cells that still contain residual ribosomal RNA. They circulate in the peripheral blood for 1 to 2 days before maturing into erythrocytes. The reticulocyte count is a key laboratory indicator of bone marrow activity and is especially useful in evaluating anemia. Regulation of erythropoiesis. Erythropoiesis is tightly regulated by tissue oxygen levels. Hypoxia is the primary stimulus that increases red blood cell production. In response to hypoxia, the kidneys release erythropoietin. Adequate levels of iron, vitamin B12, and folate are also required, along with the influence of hormones and cytokines. Erythropoietin, or EPO, is produced mainly by the peritubular cells of the kidneys. It promotes the survival, proliferation, and maturation of erythroid precursor cells. EPO levels increase in hypoxic conditions and decrease in chronic kidney disease, leading to anemia. Requirements for normal erythropoiesis. Normal red blood cell production requires several essential components. Iron is necessary for hemoglobin synthesis. Vitamin B12 and folate are required for DNA synthesis. Amino acids are needed for globin chain production, and a healthy bone marrow is essential for effective erythropoiesis. Disorders of erythropoiesis. Disorders of erythropoiesis may result from nutrient deficiencies, bone marrow failure, or increased red blood cell destruction. Examples include iron deficiency anemia, megaloblastic anemia, aplastic anemia, and hemolytic anemia. Each disorder presents with characteristic laboratory findings. Laboratory evaluation. Laboratory evaluation plays a crucial role in assessing erythropoiesis. Key tests include the complete blood count, red cell indices, reticulocyte count, iron studies, vitamin B12 and folate levels, and bone marrow examination when indicated. Summary. In summary, erythropoiesis is a vital physiological process responsible for red blood cell production and oxygen transport. It is regulated by erythropoietin, nutrient availability, and bone marrow function. Understanding erythropoiesis is essential for diagnosing and managing anemias in the clinical laboratory. If you have any questions, please review the material again and relate it to laboratory findings and clinical cases.